Mr. Chairman, dear colleagues, today I will be presenting cancer stem cells as a biomarker in the preclinical diagnosis of pancreatic ductal adenocarcinoma. As you all know, pancreatic ductal adenocarcinoma, or PDAC, is the commonest type of pancreatic cancers, in which ductal cells in the pancreas grow in an uncontrolled fashion. It is associated with several clinical characteristics, which makes this cancer a deadly disease. According to global cancer statistics in 2018, pancreatic cancer has been identified as a disease in which the incidence rate equals to that of mortality rate. And the reason for this increased mortality is due to the unfavorable prognosis and low survival rates seen among the patients. And in fact, a recent study by Rahib et al. Has shown that pancreatic cancer will surpass breast, prostate, and colorectal cancers and will become the second leading cause of cancer-related deaths by 2030. The reason for poor prognosis is actually related with late diagnosis of this disease. According to a model which was published in Nature, approximately 10 years passed between the initiation of the pancreatic tumor and the generation of parental tumor clone, which generates metastatic cancer cells. And then the acquisition of metastatic capacity takes another 5 years, and the patient's death occurs 2 years after. And currently, the PDAC is generally diagnosed at a stage when the cancer has already metastasized. So in order to promote patient survival, it is important to detect pancreatic cancer at early stages, which indicates an important role for the early diagnosis. Currently, several biomarkers have been implicated in the diagnosis, prognosis, and prediction of pancreatic cancer, but the only FDA-approved biomarker for PDAC is carbohydrate antigen 19-9. It is used as a biomarker for early detection and prognosis. However, its use for general screening has not been recommended, since it can show variation with disease stage and may give false positive results. Currently, the only created treatment for PDAC is surgery, but only a very small population of patients present with resectable tumors. And high metastatic capability is a native feature of PDAC, which suggests that tumor cells metastasize early during tumorigenesis, and they continue to evolve at distant sites independently. So with surgery, even if you remove the tumor, you may not be able to prevent the development of potential metastases arising from the early disseminating cancer cells. And this means that even those patients who receive surgery can already have subclinical metastases. And among those early disseminating tumor cells, cancer stem cells play an important role in the development of metastasis. Pancreatic cancer stem cells were discovered in 2007 by 2 different groups in Germany and in the United States. And since then, several studies have been conducted to characterize those cells. And among those characteristics, their ability to give rise to all types of cells found in pancreatic cancer has led to their classification as tumor-forming cells. So therefore, pancreatic cancer cells are regarded as the seeds of the cancer. These cells are associated with several clinical hallmarks, and one of them is chemoresistance. It is mainly mediated through the expression of ABC transporters on the plasma membrane, which can effectively remove the cytotoxic drug within the cytoplasm to the cell's exterior. And pancreatic cancer stem cells overexpress these transporters, which results in a highly efficient efflux pump system compared to non-cancer stem cells. Therefore, the drug is completely eliminated within them. Another characteristic is their ability to metastasize. Cancer stem cells leave the tumor site, enter circulation, and they home to distant organs such as the liver, the lung, and the bones, where once the conditions are favorable, they promote tumor generation. But it is important to note at this stage that the circulating cancer stem cells are different from circulating tumor cells. They are present in the circulation at a very, very low percentage, which actually makes their identification, their isolation, and their study very challenging. And finally, pancreatic cancer stem cells play a role in disease relapse. These cells can divide to generate pancreatic cancer stem cells, but they also divide to generate other types of cells found in the tumor. So if you cannot eliminate pancreatic cancer stem cells completely, there is always a risk for the development of pancreatic cancer. So the aims of this project are therefore threefold. The first one is to detect pancreatic cancer stem cells in blood as a liquid biopsy for PDAC, and secondly, to detect the relapse of PDAC after curative surgery, and finally, to detect preclinical PDAC in a cohort of patients with new-onset diabetes. New-onset diabetes patients form a high-risk group to develop PDAC. While the prevalence of pancreatic cancer is relatively low in the normal population, it can rise up to 1 in 150 among patients with new-onset diabetes. And this relationship between the cancer, pancreatic cancer and diabetes has led to a bidirectional association between these two entities. This means that while diabetes for over a long time can induce pancreatic cancer, it can also be induced by the pancreatic cancer itself. So this figure here shows the time course of the diabetes and the pancreatic cancer. And an important feature of new-onset diabetes-associated pancreatic cancer is the paradoxical weight loss among patients, which become evident before the diabetes onset. And then diabetes manifests many months before the symptoms of the cancer becomes evident. So if you diagnose new-onset diabetes with no cancer-related symptoms involvement, then it represents a good model to actually diagnose cancer at the preclinical stage. Therefore, our major aim is to be able to diagnose circulating cancer stem cells in these patients as a high-risk population that we could which could actually play a critical role in the preclinical diagnosis of pancreatic cancer. So in the second part of this presentation, I will talk about the project, the technique we use, and the results we've got so far throughout this study. This study is a big-scale study which is supported by the Scientific and the Technological Research Council of Turkey with a research budget of approximately 2 million Turkish liras and an overhead budget of nearly half of it. Other than Koç University Hospital, it is supported by several other state and private hospitals in Istanbul, which contribute with the recruitment of patients diagnosed with new-onset diabetes. Up until now, we have recruited 506 patients, among which 62% has been admitted to the study, 27% refused to participate, and 11% was not eligible to take part in. When the patients arrive at Koç University Hospital, they are directed to the clinical trials unit, where they are greeted by a research nurse who gives information about the project. If they agree to participate, then they are asked to sign a consent form. Thereafter, they donate their blood, and then they are directed to the radiology department for their MRI scan. The results of the MRI scans are shared with the patients, and all the data are archived in the clinical trials unit. The research nurse then plans the dates of the follow-up visits for those patients. So following all this procedure, the total number of blood samples that we received and analyzed has reached 668. In this study, we have two major cohorts. The first one is the test cohort, which consists of patients diagnosed with PDAC. And the aim is to detect the relapse of PDAC following surgery. And the second group consists of patients with new-onset diabetes who are over 50 years of age and do not have pancreatic cancer. And the aim of this cohort is to diagnose PDAC at a preclinical stage through the identification of cancer stem cells in blood. And in the meantime, if a patient in the validation cohort develops pancreatic cancer, then he or she is transferred to a new cohort named as validation 2 test cohort. So in order to be enrolled in this study, the patients in each cohort have to fulfill the eligibility criteria before they donate their blood. And importantly, the MRI scan is compulsory for the patients in the validation cohort to confirm the absence of PDAC. And then the follow-up visits take at every 3-month intervals to look for the presence of circulating cancer stem cells in their blood. The analysis, up to date, there have been so many different kits and methods developed to isolate circulating tumor cells from whole blood. But the method which we employ does not include any of these methodologies, since, as I have also mentioned earlier, pancreatic cancer stem cells are different from circulating pancreatic cancer stem cells are different from circulating tumor cells. They are a distinct entity in the way that even one cancer stem cell can produce tumor cells. And they are present at very, very low percentages in the whole blood, so it is not quite possible to detect them with commercial kits. So the method which we employ is based on a technique that actually looks or investigates the intracellular autofluorescence specific to cancer stem cells. This method was established in 2014, and it could identify cancer stem cells in diverse human solid tumors. And it is based on the presence of specific vesicles that is present within the cytoplasm of cancer stem cells. The role of these vesicles generally include maintenance of stem-like properties such as chemoresistance, but they can also have a role in the storage of riboflavin, which is vitamin B2. And riboflavin is a fluorescent molecule. So the question is if we can identify circulating cancer stem cells in peripheral blood through the characteristics of intracellular autofluorescence. And for this we use flow cytometry.
Doktor Tunçer, toparlar mısınız?
Tamam, az kaldı. And for this we use flow cytometry, in which the cells isolated were treated with riboflavin and then submitted to flow cytometry for analysis. And those autofluorescent cells, they appear on the top right-hand side of the dot plots as illustrated. So from now on, I will ask you to concentrate more on that part of the dot plots illustrated in the coming slides. So as the final part, I will show you some data from our studies. So this is the analysis of peripheral blood samples from a healthy donor and patients with PDAC and new onset diabetes. And the analysis after riboflavin staining has indicated the presence of autofluorescent cells in the patient with PDAC, whereas in new onset diabetes, we did not detect this, which was the case in most cases that we observed on the patient's first visit. We then went on to investigate the presence of autofluorescent cells in the tumor tissue and have identified them both in the absence and presence of riboflavin. And when those isolated cells were investigated under fluorescent microscope, riboflavin has been shown to accumulate within the cytoplasm. The cancer stem cells were further characterized by their ability to form spheroids under specific cell culture conditions. They expressed specific cell surface markers, and also they had higher levels of pluripotency-associated gene expression as indicated by black bars in comparison to pancreatic tumor cells. And finally, I will show you a few slides about the specific cases that we identified in this study. So this is the peripheral blood analysis for patients with new onset diabetes on the day of diagnosis and in follow-up visits. In patient number 1, we observed an increase in the percentage of autofluorescent cells 6 months after diagnosis, and this trend has continued 3 months later. But in another patient, we detected higher levels of autofluorescent cells both on the day of diagnosis and on follow-up visits. This may indicate a risk for the development of pancreatic cancer in these patients, which we will continue to look into in the following months. And the second case is about a PDAC patient and their analysis of peripheral blood on the day of surgery and on follow-up visits. And following surgery, we observed a decrease in the percentage of autofluorescent cells, but in another patient, similar levels of circulating autofluorescent cells were present both on the day of surgery and in the following months. So this may indicate a risk of recurrence for that patient, and again, we will continue to investigate in the coming months. And finally, we have 2 other specific cases. One of them is a patient with new onset diabetes who was diagnosed with new onset diabetes, and the analysis of the peripheral blood showed the presence of circulating autofluorescent cells. But the story of this patient is slightly different, because 1 month before the MRI scan was taken, the patient experienced some abdominal discomfort and diagnosed with acute pancreatitis. And the MRI scan then confirmed the presence of a pseudocyst secondary to pancreatitis. But the patient's complaints were persisted, and then just purely based on the evaluation of the clinical parameters, he was offered laparotomy operation, which detected extensive peritoneal metastasis, and the pancreatitis was found secondary to the tumor present in the pancreatic duct. He was offered chemotherapy and then withdrawn from the study. And finally, the last patient that we observed was diagnosed with new-onset diabetes, and on his second visit, we detected autofluorescent cells in his peripheral blood. A few weeks later, the patient informed us that he was diagnosed with lung cancer and withdrawn from the study. So, for the future work, our studies will continue with the analysis of blood and tumor tissue from the patients and further characterization of the pancreatic cancer stem cells, and all the validation studies will be conducted at the end of the follow-up period. And finally, I would like to thank everyone in the lab for their continuous support, and to Professor Mark Tarkan. And I'd like to acknowledge all the supporting institutions which contributed with the recruitment of patients diagnosed with new-onset diabetes. And this study is funded by a research grant from the Scientific and Technological Research Council of Turkey. And thank you all for your attention and for listening.